Hi, so I want to go ahead and explain a little bit about experiment number 1 before we get into it. The first thing I want to do is go ahead and expand upon the equation we have up here using words and go ahead and write the chemical formula. So we do know that we're going to have 6 of our carbon dioxide added to 6 H2O or water, and that we're going to be converting that under the influence of sunlight into glucose, which is C6H12O6 plus 6O2 or oxygen gas. Now, one of the reasons why I wanted to put this up here is because we actually have 2 sets of reactions that are taking place during photosynthesis. Now, of course, that is a simplification. There's actually far more than 2 reactions taking place, but 2 main sets of reactions. We begin with something we call the light-dependent reactions. And what we're going to notice here is that from this water, these oxygens are going to come over and form the oxygen gas. So during the light-dependent phase, we're actually using the energy from sunlight to increase the energy in electrons. We go through a couple of photosystems, photosystem 2 and photosystem 1. We produce ATP, we produce some electron carriers, which are going to fuel the second stage of reactions. But we begin all of that by breaking a water so we can get hydrogen ions, electrons, and we produce this oxygen gas at that point. The reason I bring this up, we might want to take a look for some oxygen bubbles after we've been undergoing photosynthesis for a while. So we're going to take a look and see if we can find those. The second set of reactions are going to be the light-independent reactions, where we're going to take that carbon dioxide and some of the components of water, and we're going to make that glucose. So let's talk about experiment number 1 specifically. In experiment number 1, we're going to have 4 test tubes, and you can see right here that they're going to be different. We're going to have 2 test tubes that we add some cut spinach to. Now, this is going to be what's going to be undergoing photosynthesis. We have 2 test tubes that don't have any spinach. So I want you to think about why we have particular things in particular tubes. Clearly, if we don't have spinach, we don't expect photosynthesis to occur. So why is it that we're actually including some tubes that don't have any spinach? We are going to add to all of these tubes some phenol red. We've worked with that before when we were looking at cellular respiration. Phenol red is an indicator of pH. And at lower pHs, when things are a little bit acidic, it's going to be yellow. When things are basic, they're going to be red. So we have a potential here for some color change. We're initially going to mix this up to where it's red, and we're going to add some of that to tube number 4. We are then going to blow carbon dioxide into it until we can turn it yellow, and we're going to add that yellow to the other 3 tubes. Then we can take all of these and set them out of the sun. Test tube number 2 is going to be placed within a photoblocker, so I want you to think about what that means. What is test tube 2 not going to be exposed to that all of the other tubes are going to be exposed to? So let's go ahead and set up our experiment. Okay, so I'm over here ready to set up our experiment, and you can see that I have my 4 tubes laid out in front of me. I have a beaker of water to which I need to add 4 milliliters of phenol red. I just want to go ahead and check in my lab manual. Right now I'm on pages 126 and 127 for this particular experiment. I do have my 130 milliliters of tap water. I need 4 milliliters of phenol red, and I can extract that using this pipetter here. So in order to do this, I need to go up to the 1 mark right here. You can see that I have a 0 up here, and if I fill up to the 1 on this 5 millimeter pipette, I will have 4 milliliters of solution in there. So I'm going to fill that up. I'm going to add that to my beaker here. I can just click this button to release. And if I want to, I can push down on top or use the wheel to make sure I eject all of that. So at this point, I have my phenol red, and I can mix that up just a little bit. And I think back to my tubes. One of these is going to get phenol red just as we see it right now. So I'm going to add some of that to tube number 4. And once I do that, I can cork tube number 4. And I can get prepared to add carbon dioxide to this to ensure that we have an acidic environment. So I like to do this with some white paper. It helps me detect the color change. And one thing I've learned over the years is that sometimes it's hard to detect the color change while this is in a beaker. So every once in a while, I like to pour a little bit out. And I like to take it next to my tube number 4 and hold it up to a white background. And I can see I already do have a little bit of a color change. I may produce a slight increase in that color change, but I'm already most of the way there. Okay, so I'm going to check one more time. Key thing is I need to be able to tell the difference between these two tubes, and it looks like I can do that quite well, so I think I'm ready to go. Now test tube number 3 is simply going to receive that slightly yellow phenol red. I do need to also add that to test tubes 1 and 2. I may not top those off just yet. I need to add spinach to both of these. And of course, in the interest of controlling this experiment, I want to make sure that these are going to have the same amount of spinach. So I'm going to go ahead and weigh that. I will turn on my scale. I will add my weighing dish. I will zero my scale. I'll bring some cut spinach over. Make sure that I've got 1 gram. Looks like I'm a little bit short. Okay. Looks like I'm within my 1 gram. I'm going to go ahead and add that to tube number 1. It's a fun thing about adding spinach to a tube. It doesn't necessarily want to go in there. But I can use my straw to push it in. I want as much leaf surface area exposed to the sun as possible. And I want to make sure that all of it is immersed in fluid. So I can top off tube number 1. Put a cork in that one. I can weigh out spinach for my next tube. It's like a little heavy. All right, there we go. I'll put all of this in tube number 2. I'll top off tube number 2. I'll place this in a photo blocker. I'm going to move all of my tubes towards the front end of my test tube holder to make sure they get good access to sunlight. And I'm ready to place these out in the sun for an hour so we can come back in an hour and see what our results are.